So where we kind of left off here was we started looking at the more complicated stuff here when it comes to dominance and heredity, et cetera, et cetera. So, normally when we think dominant, if I say a red flower is dominant, the dominant allele, and you breed it with a white flower, the offspring would come out red. But we also have something called incomplete dominance, where red and white gives you pink. We have something called codominance, where red and white gives you red and white. We also have an instance where we can have multiple alleles, and this can adjust things a couple different ways. It can give us more various combinations. For example, human blood, 2 alleles. We looked at how that blood, how your blood type gets chosen and how you can pass that on. We saw parents who have type A and type B blood can have a kid who has type O blood, as an example. We also looked at a hierarchy of dominance, where different alleles are more prioritized. So you have multiple dominances. We looked at so with that hierarchy, with that multiple alleles there, you could also get epistasis. This is where one allele hides the other one. In this case, you look at Labrador dogs. Even though both these genes are recessive, this indicates whether a dog is dark or not. The E's are whether it's dark or not, and the B's say what color it was. Even if it's a dominant B, if the E is recessive, it doesn't have the capacity to grow dark hair. It doesn't have that gene to be able to do it, even though it's supposed to have dark hair. And then we looked at sex determination. The mom has 2 X chromosomes and the male has XY chromosomes. And we just kind of discovered how eventually what this kind of means is that the man decides the gender for one. Man decides gender. In females, one of these X chromosomes gets deactivated, and that men are much more likely to get affected by recessive X-linked traits. And then finally, we looked at something called polygenic, where multiple things control the same thing. So there are multiple genes that affect skin color, for example. It's not just one. It's not just going to be you're either black or white. It's a whole bunch of them. And the more dominant they are, the darker the skin tone, for example. And this is true for stuff like height and eye colors as well. We try and simplify it, but it is affected by a couple different things. And then finally, we looked at environmental influences, right? We see this a lot in plants. We see this in animals. For example, in a cold region, the extremities of certain cats or certain animals will get black because black absorbs sunlight better. In winter, rabbits turn white to try and camouflage better. The genetics of it doesn't change. It's just the phenotype of it changes. What it looks like changes. It's still the same genes. And then we looked at twins and how those can be really helpful with figuring out whether something's environmental or whether something is due to genetics. So it was a lot, right? This complex for a reason. Yeah, okay. So we're going to finish this up here. And we're going to start looking at the chromosomes. So we've clued in a lot to heredity. Where we're kind of pivoting the course now, more and more to, is we're going to start looking at humans and specifically chromosomes. And this is going to come back to this concept of doing stuff accurately eventually. So we're going to look at chromosomes and how this affects human heredity. So this is 11.3. Okay. So we do not just study genes, we study entire chromosomes. And if you recall, there are 46 of them. You don't need to know how they do it, but what they just stain it and they zoom in, right? They stain it so that it's easier to see against the backlight. One important thing that we get from this, though, are karyotypes. So a karyotype is when we take all of these chromosomes. There's 46 of them, which means there's 23 pairs. I take my 23 pairs. I'm going to take out the one that does gender. I take out the one that does gender, and now I have 22 left. And I'm going to arrange them from smallest to biggest or biggest to smallest. Second. So again, karyotypes is where I'm arranging my chromosomes in decreasing size. I'll show you that, it'll make more sense. We also have telomeres. So these telomeres are protective caps on a chromosome. And I'll show you again what that looks like in a second. But basically, this contains DNA and proteins, and it serves a protective function. It's a helmet for it, essentially. Okay. There's actually not a whole lot more, to be honest with you. So these karyotypes, what do they look like? This. This is what they look like. And so there's 5, 10, 15, 15 plus, there's 7 here. So there's 22. And then here's the 23rd pair. So there are all 23 chromosomes pairs here. The difference is this, so this is 2 X chromosomes here. This is female, right? Double X. This is X and Y, so male. So again, if you're looking at the chromosomes, like kind of looking at the differences, the lengths are different. You know, these dyes just show different genes as well. We'll eventually look at how those are composed of. But you know, it's different, right? It's different. There's genetic diversity that emerges from this, of course. Obviously, though, sometimes problems can happen. The most notable problem we see, and not just in humans, we see this in animals, is what happens if I remove a chromosome, add on a 24th pair, or I add in one into one of these 2 pairs to make a triplet. So we see that a lot, specifically the last one, where instead of 2, I'll have 3 here. What complications can that make? So that process is done through something called non-disjunction. So this is when those sister chromatids don't separate properly. If you remember, when we have the cell, and we have those things that are all lined up, right? They're all lined up in the center. Here's my spindle, yada, yada, yada. What's supposed to happen is these are supposed to pull apart nice and cleanly, right? But sometimes they're stuck too much together, and both of these will go over here, and I will end up with something like this. I have 1 mono, and I have 3 tri-soms. So this entire process here is called non-disjunction. And if you even wanted to draw this part here in the bottom right, that'd probably be a good idea for this actually. Just to kind of show where that went. Instead of having two twos, there's a 1 and a 3. So the most common form of this in humans is when we go, there's a reason I circled this one down here, the 21st pair. If I add on one here, so this is male, and I've added on a third chromosome pair to this. What ends up, or sorry, not a third chromosome pair, a third chromosome to this pair. It's a trio now. You get Down syndrome. That's what results in Down syndrome. Is there's just an extra chromosome there. So it portrays itself quite a different ways. I used to actually run a home for people with Down syndrome, but there's usually some really distinct facial features, usually shorter height. There can be some mental disabilities that do happen. Heart defects, extremely common. About one in 3 babies with Down syndrome has to have a heart surgery. There's usually like a little minor hole in the heart to fix itself. Dementia is incredibly more likely as well. You don't need to know the actual characteristics, but that's what Down syndrome is. It's an extra 21st chromosome. Okay. So we are able to analyze DNA of fetuses, actually. So we can do fetal testing, and we can look at a whole bunch of things. There's a screen, and it's not just Down syndrome. There are many things that are screened for when you go through pregnancies. One of these looks at the chromosome status, chromosomal status. Okay, we're actually almost done. Actually, no, we are done. So that's it. The bulk of this really was that second part. That's where most of your effort needs to go. So today's gonna be really simple. We're not gonna start a new thing on Friday here with biology. So when we come back on Monday, we are going to, if you guys remember those Plunkett charts and pass it on heredity, that's when you're gonna do that experiment that we talked about. So that is coming Monday. It's lots of fun. You guys will enjoy it. So those of you online, your packages have either been mailed out or they're available for at the place you told me to leave them there. And if you don't get it by Monday, that's fine. You can you'll probably get it by Tuesday or something. You can do it then. Words. Oh yeah, so today, we're gonna keep it nice and simple today. There is a genetics exam I'm gonna open up on the textbook here. So this is a combination of a couple things. A lot of these are, again, exam-ish style questions. It's meant to really reach across multiple units. Once you guys have finished that, if you have those study guides still, if you can finish those, you do not need to hand those in. Those are yours. I have your old stuff I meant to give back. I will give that back to you today too. If once you finish that, you can either redo the old quizzes, you could work on more flashcards. And yes, I know we've done that a lot, but we are only at 2 weeks left. So there's actually not a whole lot of time left in the course. And if you're like, what do I do for those flashcards? There's those end of topic questions that we do for a lot of those notes. Think about those 3 questions we did yesterday, for example. In your textbook, there's lots of questions there at the end of each chapter, end of each section. There's the vocabulary, there's the study guide questions on the LMS. So there's a whole lot. There's your notes.
These assignment questions, like on the textbook, you could do flashcards.
Yes. So you could do stuff like that, for example. And yeah, and stuff like this. And there should be even an answer key somewhere, at least in the back. And if not, I have that too. If there's a question of no, like I can help you with them. Yes, yeah. Okay, so I'll let you guys get at that. The really big takeaway that we really need here is 11.2. The complex part. If you're not sure on something there, please let me know. That stuff's massive. As well as those Plunkett charts from Unit 10. If you're not sure on those as well, please let me know. Those are the 2 biggest things here.